Thank you so much to everyone for attending today. And I want to thank Adrene and other members of the Variant Effect Seminar Series for inviting me. I am a current second year resident at the University of Michigan, and I'm going to be talking to you about some of the work that I did as a graduate student at Vanderbilt University. So we're going to talk about multiplexed assays of the IKS channel complex, and I have nothing to disclose. I do want to acknowledge special thanks to members of the Roden and Glaser labs who have sort of carried on this work after my departure. These are some of the main players, and there's a lot of other people who have also contributed in many ways. So we're going to start with sort of a 10,000 foot view as to why we're going to look at the IKS complex. Heart physiology is controlled by carefully orchestrated electrical activity. And we can usually measure this electrical activity by placing probes on a patient's chest and measure something called an EKG. If you zoom into the EKG, you'll see one of these complexes. And there's an interval that cardiologists measure that can be important called the QT interval highlighted here. Prolongation in this QT interval can lead to fatal arrhythmias, and you don't need an MD to see that this EKG looks pretty different and pretty disorganized compared to the one that I showed you before. So what causes prolongation in the QT interval? One of the channel complexes, the IKS channel complex, is important for cardiac repolarization, and rare variants in the IKS channel complex, so in either KCNQ1 or KCNE1, can cause prolonged QT. So we started with KCNE1, and our goal was high throughput functional ascertainment of protein coding KCNE1 variants. The reason we started with this gene is because 77.4% of the variants in this protein are either VUS or have conflicting information. And the variants that are seen in ClinVar actually represent a small proportion of the total number of variants that are possible in this protein. So here in the blue, this diagram is to scale. In the blue, you have the variants that are present in ClinVar, in the yellow variants that are present in NOMAD, and in the green variants that are present in literature. And the white circle indicates all the possible variants in this gene. We tested KCNE1 function in two different ways. One was to test whether variants caused altered cell expression, and the second was to see if variants cause defective protein interaction with its biochemical partner, KCNQ1. So in order to do that, we created a bar-coded library, and then we conducted a trafficking assay and a fitness assay. And for purposes of time, I'm only going to talk about the fitness assay today. Just to give you a sense of what our space looks like, KCNE1 is a 129 residue protein. So we aimed for a library that included 21 variants at each position, aiming for a library of 2,709 variants. So talking about the fitness assay, one of the challenges that we ran into is that if you were to look at voltage on the x-axis and the amount of current that passes through the KCNQ1 or the IKS complex on the y-axis, when looking at the resting potential of the cells in which we use this, in which we conducted these assays, which is around -30, you'll see that there's a very small amount of current that passes through the cells. If you express KCNQ1 with its biochemical partner KCNE1, again, you have a very small amount of current conducted through the cell at -30. We looked in the literature for a gain of function variant in KCNQ1, KCNQ1 S140G, which also had a small amount of current at the resting potential of HEC cells. However, in the presence of KCNE1, this gain of function variant had a detectable amount of current at the resting potential of HEC cells indicated right here. We then validated our assay by taking a stable HEC landing pad cell line that stably expressed KCNQ1 S140G, transfected it with wild-type KCNE1 or variant KCNE1 plasmids and empty plasmids, and grew out the cell pool for 20 days. Here on the x-axis, you'll see time, and on the y-axis is the casein variant abundance compared to the empty plasmid cells. And known loss of function variants, so in this case, L51H and D76N, do not show any change in their variant abundance over the course of 20 days, whereas variants that are wild type, represented in black, or gain of function do have an increase in abundance over the course of 20 days. So with this assay in mind, we took our HEC landing pad cell line, stably expressed the gain of function KCNQ1 variant, expressed our variant library, and then did high throughput sequencing at day 0, and then grew the cell for 20 days, over which we had variants. Our hypothesis was that variants that had functional IKS channel complex would be depleted from the cell pool. So, and variants that did not have functional IKS complex would not be depleted from the cell pool. So at day 20, we did another high throughput sequencing experiment, and then took a log2 ratio of the variant counts at the 2 days to come up with our functional scores. Using this method, we were able to obtain functional scores for almost 94% of all KCNE1 variants. And here I represent the breakdown of how these variants, of how this library was represented. We saw that our fitness assay distinguished well between synonymous and nonsense variants. So here on the x-axis is function score, and on the y-axis is frequency of variance. Our synonymous variants mostly congregated around a score of 1, and then our nonsense variants mostly congregated around a score of 0, and then missense variants followed a bimodal distribution where the peaks corresponded with a peak around 0, indicating loss of function, and a peak around 1, indicating normal function or wild type like function. The next figure is going to be the heat map of the KCNE1 function assay. So on the x-axis is going to be position in KCNE1, and on the y-axis is going to be each different variant that is possible. And then in this yellow, purple, and blue ribbon, I'm representing the secondary structure of the KCNE1 protein. So this is what the heat map looks like, and there's some proof of concept. There are some proof of concept things that stick out pretty clearly. So overall structural trends. So for example, you'll notice that around the transmembrane domain, a lot of positions are resistant to variation. And then we also have these 2 intracellular domains, which also appear relatively resistant to variation. I'd also like to draw your attention to the nonsense variants at the end of the tail end of the protein that have not normal function scores, indicating that this tail is dispensable for the overall function of the protein. So this is a structural model of the protein of KCNE1 overlaid with the mean missense variant scores. And you'll notice that the transmembrane domain and 2 intracellular domains are particularly resistant to mutation. When we look at these domains in the context of the entire channel complex, we notice that these domains actually indicate potential amino acids that might be in interaction with other partners of the IKS channel complex. So for example, here, and then also here, which I've zoomed in here. So these variants in the first intracellular alpha helix are actually pretty close to KCNQ1, and we hypothesize that there's a biologically meaningful interaction between the KCNQ1 variants and the KCNE1 variants. And then this panel right here, which is zoomed in here, is the second alpha helix in the protein, in the intracellular member of the part of the protein. And these variants are particularly close to calmodulin. So we hypothesized that there was a biologically meaningful interaction here as well. Apart from looking at big structural sort of trends, MAVE data can also be used to assess individual variants. So for example, L51H is a known trafficking null variant, and it has a low fitness score in our assay. And then D76N, which is a known interaction variant, has normal trafficking scores in our trafficking assay, which I'm not showing here, but a low function score as indicated by the red here. The other thing that I wanted to sort of highlight is running complementary MAVEs on the same protein can highlight different mechanisms of pathogenicity. So when looking at both trafficking and fitness assay scores, pardon me, when looking at both trafficking and fitness assay scores, looking at trafficking scores on the x-axis and fitness scores on the y-axis, we see three different clusters. One cluster is where trafficking, the variants do not traffic well to the cell surface, and thus they do not function as well. The second group of, and I'm representing only nonsense variants here. The second group are normal in their trafficking and also relatively normal in their function scores. And these are the tail nonsense variants that I indicated to you a few slides before. But a lot of the nonsense variants have normal trafficking scores, but deleterious functional scores, indicating that there might be a defect in the protein interaction between KCNE1 and its biochemical partners, leading to a deficit in the overall protein function, which is potassium conductance. Based on studies that have been done in the past, MAVE data can be a useful way to complement the ACM, American College of Medical Genetics, American Association of Molecular Pathology criteria, or the ACMG AMP criteria, if they're a direct measure of protein function. So here on the x-axis, I have the function scores, and on the y-axis, I have the peak current, which is basically electrophysiological gold standard way of measuring the potassium conductance through the IKS channel. And you'll notice that the correlation between our function scores and our peak currents is pretty robust. The second criterion is that the MAVE or the assay should have a dynamic range of function. And what that means is that they're able to well distinguish between pathogenic and benign variants. And as I demonstrated in the past, our function assay is well able to distinguish between synonymous variants and between deleterious nonsense variants. And then it's able to distinguish, the third criteria is that it's able to distinguish between pathogenic and benign variants. So previously known pathogenic and benign variants associated with long QT syndrome or have wild type like function are going to be represented on the x-axis, and the function score is going to be represented on the y-axis. You'll notice that a lot of the arrhythmia variants have low function scores, whereas a majority of the wild type like variants have normal function scores. Based on the calibration of our assay, we were able to implement the ACMG AMP functional criteria at the moderate level for pathogenicity and at the supporting level for benign variants. The next natural progression of this setup was to extend our trafficking and our functional assay to KCNQ1, which is the larger biochemical partner in this IKS channel complex. And here is the distribution of ClinVar missense variants for KCNQ1. And you'll notice that a majority of these variants also exist in the VUS or conflicting, also have VUS or conflicting interpretations. Again, looking at the two major ways in which we assessed function for KCNE1. For KCNQ1, we also assessed alternate cell surface expression and defective potassium conductance. In order to do that, we again created a bar-coded library of KCNQ1 variants. And this time, our protein is 676 residues large. So we were looking at a library space of almost 14,000 variants. We conducted a trafficking and a fitness assay. And for the sake of time, I'm only going to talk about the fitness assay today. So if you recall from a few slides ago, looking at the resting potential of HEC cells, the current or potassium conductance at the resting potential of HEC cells through KCNQ1 is actually pretty limited. In order to obtain a biologically meaningful, not biologically, experimentally meaningful potassium conductance at the resting potential of HEC cells, we used we used two activating drugs to increase the amount of IKS at the resting potential of HEC cells. One of them, RL3, is demonstrated here. On the x-axis is the concentration of RL3, and on the y-axis is the increase in IKS, or the potassium conductance. And you'll notice that this potassium conductance peaks around 1 micromolar of RL3. Thus, we expressed wild-type construct of KCNQ1 with two activating drugs, ML277 and RL3, with time on the x-axis and the competitive index of wild-type KCNQ1, so the relative abundance again, on the y-axis. In the presence of activating drugs, we had a greater decrease in the proportion of wild-type-like cells. However, when we added wabane, which affects the homeostasis of potassium in HEC cells, we were able to see greater selection for functional KCNQ1 channels. Thus, we took our HEC landing pad line again and expressed the KCNQ1 variant library, and then grew these cells in the two activating drugs plus wabane, obtained a sample at day 0 for high throughput sequencing. Over the course of 28 days, the functional variants were depleted from the total cell pool, and then we conducted sequencing again at day 28 and took the log 2 of the ratio between day 28 and day 0. In terms of in terms of the fitness assay, we were able to obtain function scores for almost 87% of all possible KCNQ1 variants. And this is what the breakdown of those variants looks like. This is a functional heat map of the KCNQ1 variants. So just to orient us again, on the x-axis is the position in the protein KCNQ1, and on the y-axis are the different mutations that are possible. In this yellow band, I'm again showing the secondary structure of the KCNQ1 protein. Again, this heat map demonstrates important structural domains, and you can see that a lot of the structural domains that end up forming the transmembrane helices are actually resistant to hydrophobic mutation. And then the P loop, which is the pore sensing domain, is resistant to mutation in general. There were also certain other motifs that became important. So the LEL motif, which is here, is important for cellular localization. In our trafficking assay, we saw that this mutation of this motif caused a loss of trafficking of the variants. Here, we're still trying to clean up our library a little bit and get more replicates. So while the data are not complete, you can notice the red, which is loss of function variants, sort of bleeding through for the variants for which we do have data in this domain. The second domain is the PY ubiquitination motif, and loss of function variant and variants in this domain abolish ubiquitination, and ubiquitination is a mechanism by which the protein is degraded in the cell. So once you abolish the degradation, you get an increased amount of protein and thus an increased amount of function in the cell. The trafficking and function scores do highlight slightly different mechanisms as well. So this is the functional heat map that I was demonstrating to you earlier. And my computer just froze for a second. I apologize for the technical difficulties. Pause my screen share. Interview. I appreciate your patience, everyone, and thanks, Laura, for the chat. Okay. Great. This computer is also as sleep deprived as I am as a second year anesthesia resident. So there we go. Okay, so here is the function heat map, and you'll notice that there's again the motifs that I talked about, and then here is the trafficking heat map. And the differences are subtle. I'm going to flip back one more time. I know flipping back and forth does give me a headache, so I don't want to induce headaches in anyone else. But again, here is the functional heat map, and then here is the trafficking heat map. So some of the big things to notice is that there are some gain of trafficking variants that we see in the proximal to the cellular localization motif that we don't necessarily see in the functional assay. And then the ubiquitination motif is actually more strongly gain of function than it is gain of trafficking. And then some other interesting trends for loss of function. So for example, in this S6 helix, we see that it is pretty resistant to overall variation for the function of the protein, whereas it is pretty resistant to hydrophobic variation for the trafficking of the protein. Next, we saw that MAVE scores correlate with control variants with low sensitivity but high specificity. So on the x-axis, I have benign and likely benign variants. Benign and pathogenic variants, and on the y-axis, I have trafficking scores. You'll notice that most of the benign variants sort of congregate at trafficking score of around 1, whereas most of the pathogenic variants congregate at a trafficking score of around 0. This trend looks a little tighter in some ways when looking at function scores. So what do I mean by that? Again, we have benign and pathogenic variants on the x-axis and the function score on the y-axis. Most of our benign variants have a function score of around 1, and we have fewer false negatives, so fewer pathogenic variants with normal function scores compared to trafficking scores, indicating again that some variants are causing pathogenicity by disrupting trafficking to the cell surface, whereas some variants are causing defect in the potassium conductance, leading to their overall pathogenicity. In terms of calibration of the assay, we saw, again, using the same criteria as mentioned before in Brinich et al., we saw that our functional scores were able to provide evidence at the strong level in the ACMG AMP criteria, whereas our the odds path for our benign variants was only able to provide benefit evidence at the moderate level. Furthermore, we looked at variant frequency for low function score variants in known published arrhythmia cohorts versus variants in NOMAD. And we found that low function score variants, so again, on the x-axis, we have trafficking scores, function scores, and alpha missense scores. And on the y-axis, we have odds ratio of variant frequencies present in cases versus controls. We saw that low function score variants are 200 times more likely to be present in arrhythmia cases than in presumed NOMAD controls. So hopefully I've convinced you that MAVES of the IKS channel complex facilitate clinical interpretation and highlight biology. They identify important functional domains. They supplement the ACMG AMP functional criteria, but there are certain limitations. Particularly for the KCNE1 dataset, we had a limited truth set of pathogenic and benign variants. We also, given that we are conducting these assays in hex cells, we're doing them in a non-native cellular context. Obviously, these assays don't account for incomplete penetrance. Usually in a cell, there are 2 copies, but we are working with hemizygous expression. And then the KCNQ1 trafficking and function assays are actually lacking KCNE1 due to experimental difficulties. And then our future directions, we are increasing the replicates and coverage for the KCNQ1 functional assay, and then validating our function assay against gold standard methods. I do want to conclude with a case in the last 13 months of residency. So I'm a relatively junior resident, and I've already had at least one patient who had a genetic variant. It wasn't in KCNQ1 or KCNE1, but it was in a cardiovascular arrhythmogenic gene, because of which we started with a standard anesthetic plan. And this patient did not respond well to this anesthetic plan and developed arrhythmias. And as a result, we had to switch our anesthetic plan for this patient. And I've only been doing, so my first year, I didn't do a lot of anesthesia, but over the last 2 months, I've been doing anesthesia. And I've already seen how the data that the AVE Alliance is producing is actually making a difference in the lives of patients. So despite the fact that these data are very cool by themselves, the ultimate goal is to help all the patients. So with that, I would like to thank the village that basically helped generate these data. And at this point, I'm happy to take any questions. Thank you so much.
Thank you, Ayesha, for your wonderful talk. So let me check if we have questions here. We have one in the QA. So Kathy Partington is asking, how were you able to perform the electrical measurements in a multiplexed way?
Thank you so much for your question, Katie, and thank you for the chance to elaborate. So we're not doing, the multiplexed assay is not measuring direct current that's passing through the ion channel, but we're using sort of the cell death as a surrogate for what the electrical current actually looks like. So in this case, let me just pull up that slide here. So in this case, for example, our function score is not measuring electrical currents at all. This is for KCNE1, but these are individual patch clamp, one variant at a time, sort of the traditional slow throughput way of measuring electrical current, which we did for validation in this study. And we also took data that were previously available from the literature. So we're just using our function score as a surrogate. We don't have the capacity to measure electrical current at a protein-wide level. There are some technologies that are available that allow you to patch clamp like 96 cells at one time. And I can talk to you offline about that technology. We've used that for other variants. We and others have used that for other variants for electrical validation. But in this case, we're actually using the cell death as a surrogate. Hope that answers your question.
Thank you. Then I have some questions also from the organizers. I will try to reformulate a bit because I also have some of them. So if you want to relate your scores with pathogenicity, would be you have to consider both scores, trafficking and function, or you maybe suggest to only be focused on the functional score rather than in the trafficking one?
Yeah, that's a good question. So the way I see these assays is that if you look at the function score, that is going to capture variants that are not getting to the surface. Right? So the function score is taking into account if the protein is degraded, if the protein is not getting to the cell surface, if the protein's getting to the cell surface, but there's a problem in the pore or in the interaction with other partners. It's sort of capturing all of those, it's encompassing all of those mechanisms. So in order to ultimately determine pathogenicity, I would trust function scores more. But in order to get at the underlying mechanism of how a particular variant might be causing that pathogenicity, I would look at both the trafficking score and the function score. I hope that answers the question.
Okay. And then regarding the trafficking assay, can you give more details about how you measure it?
Yeah, absolutely. So I think I ended up removing those slides for my talk, but let me go back to this slide right here. Okay, great. So you have all these you have all these like protein complexes that are existing at the cell surface. What we do is that we actually had an HA tag, which is sticking. That's a very bad drawing of a tag, but we had an HA tag in the KCNE1 protein, and we have a similar MIC tag in the KCNQ1 protein. So this tag is sort of hanging out at the surface. And then we used anti-HA antibodies that were connected to a fluorophore. That star is my representation for a fluorophore. And then based on the amount of cell expression, you can imagine that the fluorescence from this fluorophore was less or more compared to variants that based on variants that had high cell surface expression versus low cell surface expression. We then used flow cytometry to separate the groups out into 4 different groups, and then took the relative. So like here, let me put this slide up. So we separated the groups out based on cell surface expression into 4 different groups, and then looked at the relative frequency of variance in each of the groups and took a ratio of that to determine our trafficking scores.
Great, very clear. And regarding this trafficking score, the fact that a variant has a high trafficking score but a wild-type-like function, what does it mean? That this variant is reaching the plasma membrane faster, there are more molecules of this variant at the plasma membrane, or what could be this explanation to this phenomenon?
So just to make sure that I understand the question completely, you're talking about a variant that has an increased or normal trafficking score, but a decreased function score. Is that correct?
No, I think I saw that some of the variants would have an increase in trafficking, but a wild type like function score
Ah, I see what you're
or
saying.
something like that.
Yeah, so the this is one of the things about that the first speaker also mentioned, where sort of measuring a dirty phenotype, right? So my hypothesis would be that there might be the functional sort of let me just pull up this guy right here. So when this guy gets up to the surface, there are a lot of things that can sort of go wrong in terms of affecting function, right? So there can be defects in interaction between KCNQ1 and KCNE1. There can be problems in the pore sensing channel. These complexes actually accumulate in multimers, so there can be problems in that. So my hypothesis is that there might be slight differences, slight decrease in the function of one complex, but the cell is able to compensate for that by increased production.
Okay, sounds like a good hypothesis actually.
Mhm.
And well, I think last but not least is about the interaction between KCNQ1 and KCNE1. So you said that for measuring KCNE1 activity, you use this background mutation in KCNQ1. And I was thinking, is this mutation could alter or affect the interaction between these two proteins? And so the effects that you are measuring in the mutations can be somehow misinterpreted or something like that.
Yeah, that's also a good question. So one of the things, and I'm sorry I don't have it marked here, but S140G is actually when we looked at it in the context of this complete structure, it's actually pointing away from the part that faces KCNE1. So my hypothesis is that it does not interact with KCNE1, causing defects, causing like erroneous loss of function or gain of function. But I haven't tested that interaction experimentally.
Okay, and a last question that is related to this is
Mhm.
that if there can exist complementation between variants in KCNE1 and KCNQ1 to
Mhm.
conserve the activity of the whole complex.
If there exists, oh, I see what you're saying. So let's say we have a slightly decreased function variant in KCNQ1 and a slightly increased function
Yeah,
variant in KCNE1, and
correct,
overall
correct,
you
something
end
like that.
up with normal function.
Yeah.
Huh, that's an interesting thought. Um, yeah, it's definitely possible. And the reason I say that is because the drugs that increase KCNQ1 function have been studied to sort of abrogate or decrease some of the loss of function phenotype from KCNE1 variants. I don't know how, I don't like that. I don't know probability wise how rare that would be, because KCNE1 is such a small protein that variants in that protein are pretty rare to begin with. So if you have like two rare, and then these conditions, the rare variants that actually end up being deleterious are pretty rare to begin with. So I feel like having like two hits in these like relatively smaller proteins would be pretty improbable, but I wouldn't be surprised if it exists, particularly in like the big consanguineous cohorts that exist out there. So that would be interesting to see.
Okay, thank you a lot. Well, I think we are running out of time, so I will like to say thank you to both of our speakers in the name of the seminar organizers who had a very good meeting today, and we are looking forward to see all of you in the next seminar series in September. So thank you very much.